The talk title is Understanding the Cellular Basis of Corpus Callosum Crossing Defect in CTNNB1 Syndrome. Please welcome Dr. Arpan Parichha, researcher, Tata Institute of Fundamental Research in Mumbai.
Hi, thanks to the organizers for inviting me and giving me an opportunity to talk about corpus callosum defects. And I feel really sorry because I could not be there with you guys and interacting with you. But I would try to highlight on corpus callosum defect in CTNNB1 syndrome. I hope my screen is visible, right? Okay.
Yes, we can see it.
Okay, great. So corpus callosum is the biggest nerve tract that connects two halves of the brain. Like our left hemisphere is connected to the right hemisphere with these gigantic nerve bundles. Now, in different neurological disorders, corpus callosum defect is pretty apparent. So here you can see corpus callosum in different angles, from the side view, the sagittal view, the horizontal view, if you cut it like this, and the coronal view, if you cut the head like this. In many cases, there has been agenesis of the corpus callosum, or the corpus callosum gets thinned. So, for example, this is a case of autism where you can see the arrows point out to the kind of like incomplete location of the corpus callosum. Let me point it out for you as well. So corpus callosum defects are pretty common in many neurodevelopmental disorders, including CTNNB1 syndrome. So I think this was the first report where you can see the corpus callosum here is pretty much thin compared to the picture in the left-hand side. So several data from Dr. Damian Spela point out that there are many other patients. Wait, let me hide the floating control. Just let me. Okay, this thing is guarding, I think. Okay, so there are many clinical variants that are found in Slovenia, which had in those cases the patient had corpus callosum defect. So the radiologist report point out that there are different aspects of the corpus callosum which was not proper. Maybe it was more thin, maybe some part of the corpus callosum was missing, sometimes there was volume loss, or sometimes there is a total shortening of the corpus callosum. Now, what you can see is like basically there is clinical variance that there are XYZ mutation in the CTNNB1 gene, and there are clinical phenotypes that corpus callosum is not crossing. Now, there is a big black box in between that why the corpus callosum is not crossing. What is preventing the corpus callosum from crossing from one hemisphere to the another hemisphere? So what is the role of beta-catenin in the neurons or even the cell types that help the corpus callosum to cross? So these are the two predominant questions we would be explaining using a mouse model. And that would give you an answer that why corpus callosum is not crossing in many of these CTNNB1 syndrome kids. So this is a
Dr. Arpan, it seems like your Zoom has froze, so I don't know if you can hear me, but if you could maybe back up a titch, we lost you there for about 10 seconds.
Oh, sorry. So basically, what I was saying that in this particular diagram, you can see there is a neuron projecting from one hemisphere of the brain to the another hemisphere. Now, this is quite fine, but it's quite difficult process because it has to project and go to another hemisphere. And why it doesn't go straight downwards or backwards? Because there are cell types, there are glial cell types, which tells and guides this neuron to go to the desired location. So these cell types are kind of like a signal post. Imagine a car is going through a road, whether it goes straight or take a roundabout is dictated by the signals, right? So in our brain, when the axons of these callosal neurons project to other hemisphere, they require also signal. They need to know where they should go eventually. And these glial cell type that I would be talking about, they require beta catenin for these guidance function. Their orientation, their this kind of like signaling mechanism is really important for the corpus callosum to cross. So overall, these cells can be thought as signaling post for corpus callosum crossing. Now, in order to do that, we use a genetic strategy in mouse called tree-LoxP system. And in this case, we do a deletion of a portion of the gene. So we have two strategies. In first strategy, we deleted kind of exon 2 to the 6 of CTNNB1. The ultimate result of that is loss of beta catenin function. And that's what happens in case of all the CTN.
Dr. Arpan, your screen
So
has
we
frozen again
plot.
for about 10 seconds again.
No, I'm
So we
so
missed a
sorry.
little bit.
I'm sorry for the internet issue. I'm not at my office, so it's a problem. So what I'm saying is like we did a loss and gain of function approach using a tree-LoxP system. In loss of function, the exon 2 to 6 is deleted. In the gain of function, the exon 3 is deleted. Now, as a consequence, you can see the protein architecture has changed. It's a simulation where you can see in green, it's the wild type protein. And in pink, you can see the loss of function variant. And in the yellow, you can see the gain of function variant. Now, you have to understand this is happening in a mouse. And let's see the phenotype of a mouse. So here, we looked at an embryonic day E16.5, which mimics the very early gestation of the human brain. In this case, you can see these green signal shows the corpus callosum is crossing and connecting two hemispheres. Now look at this part, the loss of function. Can you see the crossover? You can't, because the crossover didn't happen. And you can see the same thing in the gain of function as well. So when beta-catenin is gained or lost in these specific glial cell type, the corpus callosum could not cross. And this is true for another age. Sometimes what happens in developmental biology is that things catch up eventually. But in this case, what you can see is these gigantic kind of ball-like structures are misguided, misoriented axonal fibers, which forms a probed bundle kind of situation. So it's like many cars are stuck in traffic jam, don't know where to go. We wondered like what is happening with these glial cell itself. And we were looking at these glial cell itself because they are the signaling cell to the corpus callosum. But let me show you an overview of the brain that many of the cases in control, you can see nice crossing of the corpus callosum. But in case of all the loss of function or in the gain of function, you don't see the crossing. So developmental biologists do a technique called lineage tracing, where they label these cell types and track them over the development. Now, as a result of this, I'm not going to delve into details here, but as a result of this, we know that the tree line that we are using to delete portions of our gene marks these particular glial cell types. It's complex, but these glial cell types are the guidance molecules. I mean, and they secrete the guidance molecule. They are the guidance factor which govern whether the corpus callosum would go this way or go down. So all of these cases, you can see red is a tracker that tracks these cell type, and green is the unique identifier of these glial cell type. Now, in the control, if you see, these glial cell type are oriented in a wedge-like fashion. And in the field, they are known as glial wedge. And they actually dictate the corpus callosum crossing. Also, there are another type of glia known as inducium griseum glia, that also secretes repellent molecule which push the corpus callosal axon to the another hemisphere. Now, all these cell types were absolutely messed up in the loss and also in the gain of function. Now, you don't have to know this thing in great details to appreciate that this particular wedge-like architecture is very different in this case, right? It's absolutely jumbled and disorganized. So beta-catenin actually plays a role in cell adhesion. It connects one cell to another cell via adherence junction. So this adherence function could also be abrogated, which might have led to these phenotypes. So here is a quick quantification of that kind of phenotype. I'm not going into details. But previously, very classic literature has shown that if these orientation of these glial wedge is altered, so developmental biologists do those cool experiment where they take the portion of the tissue, flip it, and put it back and show something happens. So in this case, where the orientation of the glial wedge was changed, the axons exactly moved in opposite direction with respect to the direction they should go. And if it is removed, they go straight. And these old experiment tells us how important these cell types are in context of controlling the corpus callosum's movement. Now, we found out that beta-catenin is super important for the orientation of these glial cells itself. Now, we had a problem because there are many subpopulations. Here are some cells, here are one subpopulation, here are other subpopulations. We don't know which subpopulation is actually messed up. And that is why we need newer tools and newer approaches to understand that in bit more details. Because if you don't understand that, how would we find a treatment? So we use single cell RNA sequencing datasets from the cloud. Each of these dots and colors represent different cell types. We are interested in these astrocytic or glial cell types. And we basically subsegmented these populations and try to understand, are they labeled by some sort of like unique marker or not? And few iterations later, we found out there are specific markers like aquaporin 1, which can actually differentiate them. So you can see this particular marker only marks a certain cell type. Now, using these kind of markers or molecular tools which were not previously available, we can now understand which subpopulation of the glia is affected. So let me just quickly orient you. Here are the axonal fibers marked in green because we have antibody stained it for L1 CAM. Here is one population of glia. Look, here the GW is marked, but IG is not marked. And look at here, the IG is marked, but not the GW. So our question was, which subpopulation of these glial cell was the contributor towards these phenotypes? And we eventually found out that these IG glia or inducium grecium glia requires beta-catenin, because if you see there is loss of function of beta-catenin or gain of function of beta-catenin, they are totally messed up. Not only messed up, they are more in number. Now, what would happen if they are more in number? So imagine in a roundabout, there are so many signal posts and signaling in a haphazard direction. And you are a driver who is driving a car. You would be totally confused, right? And we think that is what happens with the incoming corpus callosum. The corpus callosum really don't know where to go. And that's why they clump up with each other and form that gigantic bundle. So this is at least in mouse right now. We don't have any way to look at it in human. But at least we know these novel cell types require beta-catenin for the corpus callosum to cross. So overall, we showed LMX1 lineage actually contributes to several of these novel glial population. And both loss and gain of beta-catenin function in these glial population lead to corpus callosum defects. And there are other players in this picture also. Beta-catenin might be super important for the neurons as well. But in this study, we looked at these novel understudied glial population. And there are two roles of beta-catenin. It can either work as a transcription factor, go to the nucleus and activate several genes of the Wnt beta-catenin pathway. Also, it can work like the adhesion factor, getting many cells to adhere to each other. And we think this adherence function is really messed up in this situation, and that's why the orientation of these cell types is abrogated. That leads to the phenotype. So that is pretty much summarizing why you might see a corpus callosum crossing defect. I mean, it simply means that the right hemisphere of your brain is not talking to the left hemisphere of the brain, and it's not good for cognition, right? So with that, I would like to acknowledge my PhD mentor. Some of the part was actually done in my PhD mentor's lab. Now I am starting my own lab in a different research institute, and we would like to thank Spela and Dr. Damian for their constant support. And I'm really sorry that I could not be there in person, but thank you for listening, and I would be happy to take questions.